Hello, everyone. Welcome to PAPERI webinar series. My name is Miriam Youssef. I will be today's moderator. Thank you for joining us for our session on Bridging the Gap, Implementing Pharmacogenomics in Diverse Population. I'm delighted to introduce our esteemed speaker, Professor Dr. Roseann Donnelly and pharmacist Nicole Keuler. Dr. Roseann Donnelly is an associate professor of pharmacy practice at Massachusetts College of Pharmacy and Health Sciences in Boston. She's also the co-founder and lead clinical pharmacist for the Pharmacogenomics Clinic at Birmingham and Women's Hospital in Boston. Dr. Donnelly is a board-certified pharmacotherapy specialist, an elected fellow of the American College of Clinical Pharmacy, and a nationally recognized expert in the field of pharmacogenomics. She collaborates with members of the International Clinical Pharmacogenetics Implementation Consortium to create evidence-based clinical practice guidelines that describe how to use pharmacogenomic test results to optimize medication therapy. Nicole Keuler is a pharmacist, researcher, and educator from Cape Town, South Africa. She leads the Masters in Clinical Pharmacy program presented by the School of Pharmacy at the University of the Western Cape in Cape Town, South Africa. Nothing is more rewarding to her than seeing students grow and develop. Her research is focused on pharmacogenomics, pediatrics, clinical pharmacy, rational use of medicine. Her PhD aims to develop a pharmacogenomics curriculum framework for pharmacy education in South Africa. She received a grant from the University of Missouri South African Education Program to observe pharmacogenomics clinics in June 2024 in the US and to bring back her experiences to South Africa. She is a vice chair of the Global Committee of Pharmacogenomics Global Research Network and a member of the Clinical Pharmacogenomics Implementation Consortium. She believes our patients will be healthier when we collaborate with an interprofessional team. Collaborating with international colleagues is one of the most inspiring opportunities in her career. International collaboration, interprofessional collaboration, education, and research are important pillars to successfully implement pharmacogenomics in South Africa. Please join me in welcoming both Professor Dr. Donnelly and pharmacist Nicole. Please note that we will have a live Q&A session at the end of the webinar. You can write your questions in the chat to submit your questions, and please mute your mics. Also, this session will be recorded and will be available on our YouTube channel. Professor Dr. Donnelly and pharmacist Nicole, the floor is yours.
Thank you so much. It's a pleasure to be here with you and have the opportunity to talk with you today about pharmacogenomics. So let me first share my screen and we will get started. All right, well, welcome everyone. Again, Nicole and I are pleased to present this session entitled Bridging the Gap, Implementing Pharmacogenomics in Diverse Populations. These are our learning objectives for today's session. I'm going to start out by focusing on the International Clinical Pharmacogenetics Implementation Consortium, or CPIC guidelines, and associated resources. I'll also get into how CPIC resources are used to facilitate pharmacogenomics implementation worldwide. And then Nicole will take it from there and identify challenges and potential solutions to pharmacogenomics implementation in diverse populations with a focus on Africa. So first, let's start with a little bit of background information. What is pharmacogenomics, or PGX? Pharmacogenomics is the study of how our genes influence drug response. It's the field of study that lies at the intersection of pharmacology, the study of drugs, and genomics, the study of genes and their functions. And it's all about how can we personalize medication prescribing based on an individual's genetic profile. The goal of pharmacogenomics is, by using genetic information to inform drug selection and dosing, we hope to maximize the likelihood of drug efficacy and minimize the likelihood of adverse drug reactions. So let's take a look at a specific pharmacogenomics example. We'll take, for instance, the medication codeine, an opioid pain medication. Codeine is what we call a prodrug, and it requires bioactivation to morphine in order for it to work via the CYP2D6 enzyme. And based on one's genetics of the CYP2D6 gene, that'll determine our overall CYP2D6 enzyme activity and therefore what kind of response we can expect to see if we're given codeine. So for example, some patients are CYP2D6 normal metabolizers, and if you give these individuals normal doses of codeine, their bodies can metabolize it into normal amounts of morphine, and we expect you'll get good pain relief from codeine as a result. Now, other patients have inherited non-functional copies of the CYP2D6 gene, and these patients are what we call CYP2D6 poor metabolizers. So if you give these patients the normal dose of codeine, their bodies are unable to metabolize or activate it into the morphine, and therefore we expect codeine would be ineffective in treating that patient's pain. And lastly, on the other end of the spectrum here, we have what's called CYP2D6 ultra-rapid metabolizers. These are individuals who have inherited multiple, or have, actually I should say, duplications or multiplications of the CYP2D6 gene. So they have more than 2 functional copies of CYP2D6. That leads to increased overall CYP2D6 enzyme activity. So if we give these patients the normal dose of codeine, well, they're going to metabolize that into a lot of morphine, and that can ultimately lead to an opioid overdose situation that with toxicity, severe respiratory depression, that could be fatal. So as you can see, based on one's genetic variation, that can really determine if someone's going to respond well to codeine, if it's going to work, or if it's going to have toxic side effects. So just like the CYP2D6 codeine example, we have a number of gene drug examples that have been well documented in the literature where we understand how genetic variation may impact if a medication is going to work for an individual or if that individual may be at risk for serious adverse effects. And so the challenge at hand is how do we synthesize all of this literature and take all of that science and bring it to the patient? Right? How do we take pharmacogenomics from bench to bedside? And 2 key questions that are at the heart of this issue is 1, how do you interpret a clinical pharmacogenomic test result? And 2, how do you use the pharmacogenomic test result to guide your prescribing decisions? So in order to help with these questions, we do have several wonderful pharmacogenomics resources that are available to us. And so I want to highlight a few of those here. Many of these are under this umbrella term ClinPGX, and they include PharmGKB, CPIC, PharmCat, and PharmVar. And you'll see underneath each logo a little bit about what their main focus is for each particular resource. But in today's session, I would like to focus a little more specifically on CPIC, or the Clinical Pharmacogenetics Implementation Consortium. And their main goal is to create and disseminate gene drug clinical practice guidelines. So CPIC was formed back in 2009, and they are an international consortium of clinicians and scientists from around the world who are experts in pharmacogenomics and have come together to create evidence-based, peer-reviewed, and freely available clinical practice guidelines for pharmacogenomics. This group is funded by the U.S. National Institutes of Health. Their resources are freely available on their website, which is cpicpgx.org. As I mentioned, CPIC members are from around the world. There are currently over 700 CPIC members that come from over 50 different countries. CPIC's work again focuses on creating these clinical guidelines for gene-drug associations. And what they do is they assemble a team, an expert team of authors for each guideline, again, clinicians, scientists, multidisciplinary group that have deep expertise in the gene and drug of interest. And CPIC also ensures that none of the authors have any major conflicts of interest in order to maintain the integrity of the guidelines and their recommendations. And what this multidisciplinary group does is they conduct a thorough systematic literature review for a gene-drug association and ultimately want to end up with the following. They want to end up with guidance for clinicians on how to take a genotype result that their patient may have, how do you translate that into a phenotype, and from a phenotype, how do you get to a specific clinical recommendation in terms of a prescribing decision for a medication. And in some cases, CPIC may say, you know what, we don't have enough evidence to actually make a recommendation, or perhaps standard dosing applies even in the setting of certain genetic variation, and that's helpful information as well. So they really are very much evidence-based and only make recommendations when the evidence supports it. Now, a key assumption of CPIC guidelines is that the pharmacogenomic test result is available. So CPIC guidelines do not tell clinicians when they should order pharmacogenomic testing, but rather they explain how to interpret and use existing results. Each CPIC guideline follows a standardized format. And so if you were to take a look at one, you'll see it starts out with an introduction, followed by information about their focused literature review. And then it gets into background about the gene or genes of interest and genetic test interpretation, available genetic test options, as well as incidental findings. The guideline then delves into the specific drugs or drugs of interest with background on each one, a section about linking genetic variability to variability in drug-related phenotypes, and then finally a little bit about the therapeutic recommendations and justification for those. And there's also a specific pediatrics consideration section as well, if applicable. The guideline continues with potential benefits and risks for the patient, as well as caveats, including appropriate use and/or potential misuse of genetic tests. So with each CPIC guideline, there are also a number of other very helpful resources that come with it. And one example of that are these gene information tables. And there are 4 main tables for each gene that is the subject of a CPIC guideline. So first, the allele definition table lists out all of the different star alleles for a specific gene and what variants define each specific allele. In terms of the allele functionality table, again, lists out all of the different star alleles that are known for a particular gene and has a function assigned to it. So does that allele confer increased function, normal function, decreased function, or no function for that particular gene? And then the frequency table. We know that allele frequencies vary across different ancestry groups. And so there's a reference table where you can look up this information for each allele based on the data that we have available to us. What are the predicted allele frequencies across different biogeographical ancestry groups? And then finally, the diplotype phenotype table. So basically, this table is a large Excel file that lists out every possible combination of alleles or all the different diplotypes that you could see for a specific gene based on all the alleles that have been defined so far. And then what is the phenotype that is mapped to each of those different diplotype combinations? And so that's a helpful resource as well. So where do you find all this great information? Well, if you go to the CPIC website, cpicpgx.org, you'll find a number of links at the top to a lot of different resources here. But if you were to click on the top left link entitled Guidelines, you would get a listing of all the currently available CPIC guidelines, of which there are over 25 different guidelines at this point in time. And you can search also in the search bar if there's a specific gene of interest or drug of interest to help you narrow the list. But essentially, once you find the guideline you're looking for and click on it, you'll get to a specific guideline webpage. And so, for example, we're looking at the guideline webpage here for opioids as with the gene CYP2D6, OPRM1, and COMT. And the very first link on a guideline webpage is the link to the manuscript itself for that specific CPIC guideline. And if you were to continue scrolling on this page, you would notice you'll see a link to supplemental information that goes along with the guideline, including detailed evidence tables summarizing all of the published literature that was evaluated, as well as lists of tables and figures that support the guideline. And towards the end of this page, you will also see links to those gene information tables that we just reviewed. So these hyperlinks will take you to these Excel files that give you the most up-to-date information about the specific genes of interest. So let's talk a little bit about how CPIC can help facilitate pharmacogenomics implementation with some specific examples. And so if we think about 4 main steps for pharmacogenomics implementation, starting with ordering the test, and then going into interpreting the results, followed by applying the result to patient care, and then finally documenting the result, CPIC really comes into play starting at the second step here with interpreting the pharmacogenomic test result. So within each CPIC guideline, it explains how do you translate a genotype into a phenotype. And this genotype to phenotype translation often is found in Table 1 of the guideline. And if you take a look at Table 1, you'll see this is an example for CYP2D6, what the different phenotypes are for CYP2D6. In some cases, we have what's called an activity score for certain genes, and then examples of CYP2D6 diplotypes that map back to these specific phenotypes. Now, again, for a complete list of all possible diplotypes and which phenotypes they map to, you can access that diplotype to phenotype table, which is an Excel file that has all the possible combinations. So going back to our steps here, for the next step, apply the pharmacogenomic test result to patient care. If you go into a CPIC guideline, they also have clinical recommendation tables that map specific pharmacogenomic phenotypes to specific clinical recommendations. And so this is the example table here for codeine therapy based on CYP2D6 phenotype. So you can see that we have a clinical recommendation for CYP2D6 ultra-rapid metabolizers. It describes the implication of that for the drug. So in this case, increased formation of morphine, leading to higher risk of toxicity. And then there's a CPIC recommendation of avoiding codeine use because of the potential for this serious toxicity. And if opioid use is warranted, consider a non-tramadol opioid, as its pharmacogenomics is similar to that of codeine. You'll notice that each recommendation has a classification. This could be strong, moderate, or optional, depending on the strength of recommendation and the evidence that we have to support each specific recommendation. You'll notice, just to highlight one more recommendation here for CYP2D6 poor metabolizers, we have greatly reduced morphine formation leading to diminished analgesia. And again, the recommendation here is also to avoid codeine, but because of the possibility of lack of therapeutic benefit versus toxicity with the ultra-rapid metabolizers. And again, the strength of this recommendation is strong because we do have quite a bit of evidence to support these recommendations. Now, another helpful tool that I'd like to point out is through PharmGKB, or the Pharmacogenomics Knowledge Base, and it's called the Genotype Selection Interface. So you can link out to this through the PharmGKB website, or if you were to just Google Genotype Selection Interface, it should pop up for you. And what this tool is it gives you basically a listing of all the different genes that have CPIC guidelines. And you have these little drop-down menus where you can choose which alleles of interest, or perhaps there's a patient that you have in front of you with a specific result. And you can fill in as many of these as you'd like. And so for my example, I filled out CYP2D6 with *1 with a duplication, as well as another *1 allele. And if I were to click Make Report at the end of that page, you will see that what this will tell you is that it will translate the genotype into a phenotype for you, and it will do this for all of the genes you may have plugged information in for. And then it will give you a hyperlinks to all the different drugs that have been associated with the specific gene or genes of interest, again, that you had populated in the previous web page. And so you would then be able to click on a specific drug of interest. So for example, if we're going with our CYP2D6 codeine example, if I were to click on codeine, it would then take me down to a section that outlines the specific recommendations for codeine based on the genotype result that I plugged in. And so on the left-hand side, you'll see the CPIC recommendations from the CPIC guidelines. So I clicked this little box here to drop down all of this additional information. It tells me what the genotype was that I submitted, the matched phenotype, the implications, and the CPIC recommendations. And I can also click a link to go to the full annotation of the guideline if I wanted more information. And then also this resource lets you compare across other guideline resources. We have the DPWG, or the Dutch Pharmacogenetics Working Group guidelines. So if you wanted to compare those, you could click the little plus symbol and see what these guidelines say. And then the other two columns are FDA, US Food and Drug Administration resources for recommendations for the specific genotype inputted and the drug of interest. So going back again to our steps for pharmacogenomics implementation, the final step here that I wanted to touch upon is documenting the test result. This is definitely an important step, considering that pharmacogenomic test results are relevant lifelong and can have implications years after a patient may have gotten the results. So it's important to understand where are you documenting these results, and how might they be able to be presented to a clinician who is prescribing a relevant medication to a patient, perhaps many years down the road. So CPIC, in addition to their guidelines, also provide a number of informatics resources to help with the implementation and documentation of pharmacogenomic test results in the electronic health record. And this includes implementation workflow diagrams, sample consult note language for documenting in the electronic health record, as well as sample clinical decision support alerts. And so this could look something like this, with a pop-up alert that could come up on the screen. Let's say, for example, a patient is a CYP2D6 ultra-rapid metabolizer, and the physician prescribed codeine to the patient. Then this alert could pop up on the computer screen to let them know that this patient has that CYP2D6 result, that they're increased risk of developing toxicity, and to consider selecting an alternative therapeutic agent. So this is a listing here just for your information of the different genes and drugs that currently have CPIC guidelines. You'll notice a number of different genes, a number of different drugs that span a variety of therapeutic areas. Some of these medications are very commonly used, and some are more specialty type medications. CPIC is also continually to evaluate the literature and develop new guidelines that come out each year. And they also go back to previously published guidelines to re-review the literature, look at what's been published since the guideline came out, and publish additional guideline updates if applicable. So if you're interested in pharmacogenomics and wanted to stay up to date with CPIC, there is a newsletter you can subscribe to if you were to go to blog.clinpgx.org and hit subscribe. You'll get on an email list, if you're interested, that will keep you up to date with what's happening with CPIC and their guidelines and other pharmacogenomics related projects. If you're also new to the field of pharmacogenomics, or perhaps you have students who are working with you and wanted to wondering about some resources to learn more about pharmacogenomics, one freely available resource is here on this slide, the Pharmacogenomics Learning Series. If you were to capture this QR code here or go to the website at the bottom here, this is a series of online modules that are interactive that help go through the basics of pharmacogenomics and the resources available, specific drug-gene pairs, as well as clinical implementation principles. And so it's a nice primer to help give you an introduction to the field as a whole. One other resource that I wanted to point out to you is this open access journal article entitled Decoding Pharmacogenomic Test Interpretation and Application to Patient Care. This is an article I had the pleasure of working on with a number of my colleagues in the field, and essentially it breaks down step by step. When you have patients' results, how do you go about interpreting those results and applying that to patient care and using the CPIC resources in order to help do that. And we set it up with a very nice framework, this ABCD framework that includes different considerations to keep in mind when interpreting and applying pharmacogenomic test results to patient care. So as we've talked about all these wonderful resources and guidelines that help us bring the science of pharmacogenomics to the patient, that brings us to the real big challenge here, which is how do we bring the value of pharmacogenomics to everyone? And this is definitely an ongoing challenge, both within the United States as well as around the world, because there are a number of implementation challenges that need to be considered. And as we think about especially helping to make this accessible and valuable to people around the world, it is important to mention that we do need more diversity in genomics studies. And we know that by and large, in most genomics related studies at the present time, most participants are of European ancestry and very few are from other population groups. And this is a significant limitation, especially in genomics when we know that allele frequencies can vary considerably across different ancestry groups, different populations around the world. So if we're only studying primarily one group, we're perhaps missing out on discovering variants that might only appear in other population groups. So the first big step that needs to happen to really make sure that we can get pharmacogenomics to everyone is that we do need to increase research across the board, have more diverse participants in genomics and pharmacogenomics research broadly. And as now we focus our attention more towards Africa, I do also want to note there is incredible genetic diversity just within Africa itself. And there are a lot of some ongoing efforts that you'll hear about where we're trying to explore that sort of genetic diversity, but still, of course, more work needs to be done. And so at this time, I'm happy to transition it over to my colleague, Nicole, who again is a pharmacist based in South Africa, and she's doing some really wonderful work around developing a pharmacogenomics educational framework for students and pharmacists in South Africa. And we'll explore with you a little bit more some of the challenges with implementation and potential solutions, again, with that focus on Africa. So with that, I thank you so much for your attention, and I will hand it over to Nicole.
Thank you so much, Roseann, and thank you, Wafa, for creating this platform. I'm very excited to share a bit of my passion for pharmacogenomics with you all today. So maybe you can just confirm if you can see my slide.
Yes, we can. Okay,
great. Okay, so this is a picture of Africa. You can see it's quite colorful. We are known as a rainbow nation. So we also have a lot of genetic diversity, like Roseann referred to. So in this presentation, we're going to explore a bit of the challenges and also some potential solutions for pharmacogenomics implementation in diverse populations with the focus on Africa. So the clinical implementation of pharmacogenomics in low to middle income countries like Africa has been very slow, and there's various reasons for that. So we have poor capacity in terms of implementation, education, and research. There's a big need for funding for research, but also implementation. There's also some concerns about the clinical applicability, and we'll explore that in the next few slides. There's also uncertainty about the knowledge on the diseases and treatment genetics because of the big genetic diversity that we are seeing. The majority of the studies that has been conducted were mostly in developed countries like the US and Europe. There's very limited actually being done in developing countries. Less than 5% of the PharmGKB database that Roseann also referred to is actually done in African population. And 15 out of the 300, this is a bit of an old reference, because there's more than 600 FDA or medicines on the FDA list with pharmacogenomics recommendations. But 15 out of the 300 in that time was actually recommendations and relevant to South Africa. So if we look at sub-Saharan Africa, it accounts for a quarter of the global disease burden, which is quite a lot. And in South Africa specifically, we face a quadruple burden of disease. And when we look at these conditions, pharmacogenomics can be relevant to retroviral disease, tuberculosis, but also a lot of non-communicable diseases. There has been some studies done in Africa, but it is very limited. So there's been some studies done in South Africa, in Zimbabwe, and especially Nigeria. There's also been some published case reports focusing on ADR reporting in Africa, and they have found that some of these are actually linked to pharmacogenomics, to certain genes that patients have, and these includes like efavirenz and isoniazid. So luckily we have our guidelines have changed to not include efavirenz as first-line treatment for ARVs, but it is something that has shown that pharmacogenomics has an influence in the treatment and the side effects that these patients experience. In Africa, we also have a lot of patients with using traditional medicine and herbal medicines, and these has also been shown to that it can lead to phenoconversion. So that is actually when there is like a change in the patient's phenotype due to medicines that they are actually taking. But phenoconversion can also be sometimes because of other reasons as well. But just considering all of this, the pharmacogenomics has a great potential to improve patient outcomes in Africa. In this picture below, you will see this was done in 2022. There was a study conducted with various stakeholders, and those countries that had a light yellow bubble has actually planned to do clinical implementation of pharmacogenomics. This is just to show you, it's from 2020, but it's just to show you the type of pharmacogenetics data and studies that has been done. So most of this was done in North Africa at that stage, focusing mostly on cardiovascular diseases and some of the infectious diseases as well. This is just a picture. This was a recent study published earlier this year, just showing you visually actually focusing on specifically CYP2D6, the proportion of the patients based on results and data that they have. And you can see Africa and sub-Saharan Africa is actually accounting for very little of the data. From the same publication, this is quite a busy slide, but this just shows you on the left the medication names, the genes that are applicable. So these are clinically actionable pharmacogenes. So this tells us there is enough information to actually make recommendations for implementation to, for example, change a dose or to change the medication. So this green here tells us these are patients that are normal metabolizers. The red is telling us patients who are intermediate metabolizers, and the yellow poor metabolizers. So this is also based on predictions on the African population. And on the right-hand side, you have the CPIC recommendations, so the guidelines that Roseann referred to. The blue is patients that would require like standard dosing, and then the orange is telling us maybe these patients need like, or they actually recommend a change in dose or a change in the medicine. So you can see that there are some medicines that would require a consideration in change of medicine or change of the dose to optimize the patient's outcome. This is to show you some projects that's happening in Africa. This is quite exciting. There's quite a lot of projects going on, various diverse fields. Earlier this year, I also attended the International Society Study of Xenobiotics in Johannesburg earlier this year, and there was quite an interesting study. It's not been published, and I think there's a lot of more studies that is available which is not just published. So the study that they spoke about was specifically done in Nigeria, and patients with sickle cell disease, and they actually did a pharmacogenomics. They implemented a project where they had a small panel to do testing, and they focus a lot on pain management. So there we look at CYP26, like the example like Roseann also used, and they did the testing, found interesting findings, and it was very interesting to see what they were actually capable of doing. So patients would get like their results on an app on their phone. So it is telling us it is possible in Africa. So in the next few slides, I want to explore a bit of the challenges with you. So in South Africa and Africa, we do not really have electronic health records yet. It is very scarce. There are some private hospitals that actually do have electronic health records, but this is really important in terms of continuity of care when we do pharmacogenomic testing. Like Roseann showed you, this is usually incorporated in developed countries into the electronic health system, and the doctor will get like a pop-up alert, and everyone in the medical team that has access to it can see that. It's also very valuable then to do monitoring and evaluation of projects. There's also some concerns about digital security, but in our setting, this is a challenge that we need to consider. Then pharmacogenomic testing can be costly. In South Africa that I know of, you can get a test for under 5,000 Rand, and yes, it is once in a lifetime test, but there are some patients that cannot afford it. There's also a lot of testing labs that is available. Not all of them are also always regulated, so that is something to consider. There's a big need for clinical pharmacogenomics education for healthcare professionals. It's not standard included in our curriculums for healthcare professional training. There's also a need for standardization, actually globally, in terms of learning and teaching, because they've seen that, when once healthcare professionals are educated, they're confident, and they can lead implementation. Then I've shown you that there's a bit of limited research, but there are some very great organizations that are actually doing research in the field, specifically in Africa and South Africa, so we're all making progress. Because of the scarcity of data from Africa, there are some concerns about the clinical validity, and that requires some more investigation and some more research, because we're not sure if our population always have the same functions, so that requires research. There's also a need for regulation, so we need policies in this setting to really govern pharmacogenomics implementation. There's a lot of ethical considerations to actually consider. Pharmacogenomics also has a great role to play in pharmacovigilance. We know that adverse drug reaction reporting is actually underreported. Unfortunately, in our setting, we cannot really make associations yet because we do not do the testing. But I believe that when we do the testing, we can actually maybe prevent some adverse drug reaction. There's also a need for next generation sequencing, but this is costly, but it can make pharmacogenomic testing cost effective and contribute to a lot of research. I told you that there's a need for standardization in terms of teaching, but there's also a need for reporting of these results and the processes needed for reporting. I've also seen that some of each company represents data differently and their reports, and this makes it sometimes challenging for healthcare professionals to interpret these reports. There's a lot of ethical, legal, and social implications to consider. We need to have knowledge about this. Healthcare professionals need to be knowledge about this to have competence. There's also a big scarcity of genetic counselors globally. And recently I attended a webinar and there was a discussion on the scarcity of genetic counselors, especially if you have a panel that, for example, test genes that can predispose a patient to, for example, bleeding or cancer. So there you really need to get the genetic counselor involved. And there were some discussions that even pharmacists maybe need to take on some counseling of this aspect. In the Africa population, we also have a very unique culture, and we need to consider that in terms of implementation. We have to think of the jargon that we are using with healthcare professionals, but also with patients. There's a lot of religious and cultural beliefs that we need to consider, and this links very closely with the ethical considerations. We also have herbal and traditional medicine use that I shared with you earlier, and this is also something that we need to consider. In South Africa specifically, we have a public and a private healthcare setting, and there are some differences in this. And at this stage, there are some testing being done in the private setting, in our setting, but I know that there are some research institutions that are planning to do testing in the public setting, but pharmacogenomic testing should be available to public patients both in public and private settings. And I think as these testings are being done more, it might become more cost effective. Currently, it's not being reimbursed by medical aids, and this is something that would need to, where we need more research to actually show the cost benefit for specific patient populations. There are some medical aids that cover this, but it's not standard, and this is where we also have a role to play to show the value of it. So in the next few slides, I want to talk about potential solutions, what we can do to advance pharmacogenomics in the African continent. So we really need pharmacogenomics stewardship, and we need a national pharmacogenomics program to really govern pharmacogenomics implementation. We need 3 pillars, which is research, implementation, and education. And we always need to consider the ethical, legal, and social implications of this test. There's a big need for collaboration between developed and developing countries. Really, both countries can benefit. So yes, the developing country benefit because of resources, because of knowledge sharing, training opportunities, but the developed country also learns about especially rare diseases that is quite unique to some specific population. And there has been some partnership and collaborative research, but I think we need more of that. So to really make pharmacogenomics implementation successful, we need stakeholder engagement, and this includes from the research to the end user, which is our patients. And we need to collaborate with industry that's actually doing these testing, healthcare institutions that's implementing the testing, research institution that can be training or doing research in pharmacogenomics. And we also need the community's perspective and the patient as well. We also need an infrastructure to build this implementation. We need very good digitalization and various of the infrastructure challenges that I've shared with you. We also need community engagement. At a conference that I attended earlier this year, there was a big discussion on community engagement of patients and getting the patient's perspective. It is ultimately their results, so it needs to be returned and they need to be educated, especially to ensure continuity of care. We do not just need international collaboration, but we also need intercontinental collaboration between these various stakeholders and just the regions, because even in various regions, there can be genetic diversity. Then we also need education for healthcare professional students, but also the patients and the public. And each of these groups really require a different approach. Then, which is very known in the literature, is that you need a local pharmacogenomics champion to advance pharmacogenomics implementation, and these are usually our healthcare professionals like our doctors and pharmacists. So yes, we do have challenges in low to middle income countries, which are quite unique to our setting, and not everything has been explored yet, but I think it can be possible if you have a roadmap for implementation, if you have a pharmacogenomics steward program to really guide implementation. And we need context-specific research, education, and implementation, because it will look very different in our setting compared to developed countries. If you would like to learn more about pharmacogenomics, this is an introductory tutorial that was published by the Pharmacogenomics Global Research Network. You can scan this QR code. This is freely available. It really takes you step by step how to work up a patient case and gives you a good summary of pharmacogenomics in cardiovascular diseases. Then the Pharmacogenomics Global Research Network. This is a global organization that aims to advance research globally. They have webinars every second week. I have learned a lot about it just by attending these webinars. You can follow them on LinkedIn, and they also have, you can join them. I've also pasted the link to their website for you. They are quite a very active group, and they have various special interest groups, so I think they really cater for anyone. The membership is free for low to middle income countries, and I would recommend that you explore this. They also have conferences, and they have the conferences here combined actually with CEPIC. Then last year, I was able to travel a bit to the US, observe some of the pharmacogenomics clinics, but then I was also able to attend the CEPIC conference. This was a very exciting opportunity for me, being in a room of experts globally on pharmacogenomics, the way they discussed research, implementation, and education. And it was also the first time that I actually met Roseann, although we have been working together for a while. And so this has really been a great opportunity for me to meet experts in the field. I also presented just my PhD in terms of the proposal and used this as a recruiting opportunity. Based on my experiences abroad, this was a publication that we published. It's also freely available, and it explains the workflow that is followed for us to think what would it look like in our setting. Then we are also having the University of Pretoria with the University of the Western Cape. We are hosting a pharmacogenomics winter school in July for healthcare professionals and healthcare professional students. We're going to talk about pharmacogenomics in South Africa, but this is really open for anyone that wants to learn more about pharmacogenomics. These are my references, and then I would love to connect with you via email or LinkedIn, and I look forward to talking with you more about pharmacogenomics.
So much, Professor Dr. Roseann Donnelly and Pharmacist Nicole Keuler, for the insightful presentation. We will now open the floor for questions from our audience. If you have a question, please use the raise hand feature or type your question in the chat. We will try to address as many questions as possible in our allotted time. So let's now start with the first question. It's here from Dr. Wafa at the chat box. It says, thank you for the valuable insights. Considering the policy landscape in many African countries, what regulatory or institutional frameworks do you believe are most urgently needed to support the ethical, equitable, and sustainable integration of pharmacogenomics into national health systems? Also, you have highlighted the importance of population diversity in precision medicine from a patient-centered care standpoint. How can we ensure that pharmacogenomic implementation respects cultural contexts, builds trust, and avoids widening health disparities, particularly in underserved communities?
Okay, so I'll start with your last question. So I think that is where, like from the patient perspective, I think that is where the community engagement really is important, and we need people to do research in that field. The community, someone told me once that the community has all the answers, but they don't always have all the resources. So they know what they want, and we need to help them as researchers and educators to actually just give them the resources and find out what they need. So I would say that we need research from a patient perspective. I'm also very passionate about the patient in this aspect because it's their results, and we need to make sure that they are comfortable with that. In terms of the framework, yeah, that is a challenging one. I think that is why we are having this stakeholder engagement workshop, because we want to have a roadmap for implementation. So it would be ideal to have a document that speaks to research, implementation, education, and to help us identify what is the most important aspect. But I think if we think of implementation, it needs to be regulated, and that's why we have governing organizations. So I can only speak for South Africa, where we, for example, have the South African Health Regulatory Authority. So they need to help us regulate that in terms of implementation. We also need educational bodies, so professional bodies that governs, for example, nursing, doctors, pharmacists, genetic counselors. So we need them to regulate the education. And then we also need like healthcare institutions and higher education institutions to govern that. Roseann, would you like to add anything to that?
I think that was a very comprehensive answer, so I don't have too much more to add. I just will echo, the community engagement piece being so important as part of the overall picture of implementation. But yeah, I think you hit on a lot of great points, Nicole.
Thank you so much for answering the question. And now we're available for our second question. If you have a question, please use the raise hand or write it. We have another question here, I think. Ms. Hannah says, thank you for the in-depth presentation. I agree that we need a roadmap or policies to direct and regulate pharmacogenomics applications in continents like Africa. So Wafa is asking, what would you say are the most immediate practical steps that pharmacists can take to begin integrating pharmacogenomics into patient care, even in low resource settings?
Yeah, that's a wonderful question. I mean, I think it really does start with education. So taking it upon yourself to seek out resources and education around pharmacogenomics. So some examples like the ones that I showed and that Nicole showed during the presentation today. There are freely accessible educational modules that anybody can do from anywhere in the world. You just need the internet connection, as well as freely available published articles that kind of guide you step by step on how you would be able to do this in practice. And then, of course, after that education piece, maybe educating your colleagues and other clinicians. And then, in order to actually implement, that very first step is, getting the pharmacogenomic data for a patient. And so that could be a challenging piece depending on where you are. Do you even have access to a clinical pharmacogenomic test? But really that infrastructure needs to be in place in order to put this into practice. So again, starting with education and then seeking out, hopefully there could be some access to some pharmacogenomic testing. And then once you have that data, going through those steps that you've learned to implement in practice. Nicole, did you have anything else on that?
No, I think that's great. I think really education, because there's a big gap and our pharmacists in practice are not always, do not have the knowledge yet because it's not integrated in our curriculum. But like you say, there are freely available resources that you can use.
I'll just add as well, going back to that question too, just looking at the CPIC website. So again, CPIC guidelines are freely available, and all of their resources for interpreting pharmacogenomic test results and applying them to patient care and all the recommendations. There's those online tools that I showed as well. So I definitely encourage folks to check that out. And even just looking at the CPIC guideline webpage and seeing what are the medications that you use in your healthcare setting, and really focusing on educating yourself about the pharmacogenomic implications of those. And so that's also a good starting point.
Thank you so much, ma'am. Thank you. Okay, so If you guys have any more questions, you can add it at the chat box or use the raise hand feature.
Roseann, there's a question, maybe this is more for you. It says, in your experience, what implementation science frameworks or model have proven effective for translating pharmacogenomics research into clinical practice, particularly in resource-constrained settings?
Yeah, no, great question. Yeah, I'm not sure. I'm not as familiar with the implementation models focused on resource constrained settings, so I'm not able to comment too much on that one specifically.
Okay, so maybe I could just, I don't have like a real model, but I think what some of the healthcare institutions have done is that they would do the testing, but as they do, they will start very small. They will start like with one gene, and then they would focus on that, and they would incorporate like a lot of like education or training for the healthcare professionals, and that is where your champion also comes in. So that has been shown to be effective, and educating your core team and including both your pharmacist, your doctor, your genetic counselors, your lab personnel. They are like your core team, and then nursing as well.
Yeah, I agree. It's a great approach. You can just, start with just that one gene. I think that makes a lot of sense. And then building from there. Absolutely.
Miss Hannah here have a question. Please unmute your mic and go ahead. Thank you.
Thank you, everybody. Here it's afternoon in Nigeria, so I'll say good afternoon to all of us. I think for me, pharmacogenomics needs to be taught in our departments, maybe genetics departments in universities, first and foremost, because it's in Nigeria here. I know it's not a field we talk about a lot. So it's something that the genetics or pharmacology departments need to integrate into the school curriculum first, and then do a lot of practicals on it, because the theory is not just enough. We need to let the students understand the practical aspects of pharmacogenomics, the tests that needs to be carried out, and the applications of the methods involved. Then if we can train a crop of students to know these rudiments, we start from there. But again, in my country, Nigeria, policies are very, very important. If not so, the scientists will go wild with their knowledge and start abusing human rights and things. So alongside, while we are educating the students, we need a consortium, a group of scientists to sit together and form policies guiding the implementation or applications of these practices. We have groups like scientific societies. Like, I'm a member of the Biotechnology Society of Nigeria. We have the Genetic Society of Nigeria. These are issues that can come up in our maybe annual meetings, to set up groups of people to sit down and start thinking up policies to guide the application of pharmacogenomics in the country. I think if we can do this across African countries, it will change the demographics from the very low percentages that we've seen. Thank you.
Thank you, Hannah. There are some researchers that are working on stakeholder engagement and some policies. So I hope that through our stakeholder engagement workshop and what we are doing in South Africa, it can also help in bigger Africa as well.
Mr. Usman, please go ahead.
Hello, good afternoon. Hello, it's afternoon here in Nigeria.
Yes, we
I
can hear
must
you.
agree. Yeah, I agree with the last speaker, even though I didn't get the name very well. In Nigeria, policies are very important. We cannot take that away. Then another thing I think very importantly is our curriculum too. That has to be looked into. I don't know how many people are interested in pharmacogenomic courses. We have to have a framework that we need to bring in people that will be interested in these courses, most especially pharmacogenetic, master's levels, and the rest. But for now, most of us are interested. Personally, I'm interested in pharmacogenomics. But the thing is that there is not any policy. I can't lay my hand in any policy or any. I don't know if they're out there. Maybe I have not met any. I have not seen any university with a pure pharmacogenomic master's degree and the rest. So that's my question. I think the framework in Nigeria would need to take it serious. And the most important thing is the policy. Number one thing is the policy. I wrote on the chart that the policy first, then the education. Then so I think somebody responded and he was saying that there are some policies that sometimes they may even stop the education, but I don't know. But this is my concern. The policies first before any other thing. Thank you.
Okay, so thank you so much again, Professor Dr. Roseann Donnelly and pharmacist Nicole Keuler, for answering the questions and for your excellent presentation. It was a pleasure to have you with us. Thank you all for attending. We hope you found it informative and enjoyable, and please have a great day.
Thanks, everyone.
Thank you.
Thank you, ma'am. It's a pleasure to meet you. Thank you.
It's a pleasure. Thank you.
Bye-bye.
Bye.
Thank you so much. Bye-bye.